Prey distributions are often clumped, and predators respond by looking for patches where prey is dense and then searching within patches. 25. Where food is found in patches, such as rare shoals of fish in a nearly empty ocean, the search stage requires the predator to travel for a substantial time, and to expend a significant amount of energy to locate each food patch. 33. For example, the black-browed albatross regularly makes foraging flights to a range of around 700 kilometers, 430 miles, up to a maximum foraging range of 3,000 kilometers, 1,860 miles, for breeding birds gathering food for their young. 34. With static prey, some predators can learn suitable patch locations and return to them at intervals to feed. 33. The optimal foraging strategy for search has been modeled using the marginal value theorem. 35. Search patterns often appear random. One such as the levee walk, that tends to involve clusters of short steps with occasional long steps. It is a good fit to the behavior of a wide variety of organisms including bacteria, honeybees, sharks and human hunter-gatherers. 36-37. Assessment. 7-spot ladybirds select plants of good quality for their aphid prey. Having found prey, a predator must decide whether to pursue it or keep searching. The decision depends on the costs and benefits involved. A bird foraging for insects spends a lot of time searching, but capturing and eating them is quick and easy, so the efficient strategy for the bird is to eat every palatable insect it finds. By contrast, a predator such as a lion or falcon finds its prey easily, but capturing it requires a lot of effort. In that case, the predator is more selective. 28. One of the factors to consider is size. Prey that is too small may not be worth the trouble for the amount of energy it provides. Too large, and it may be too difficult to capture. For example, a mantid captures prey with its forelegs and they are optimized for grabbing prey of a certain size. Mantids are reluctant to attack prey that is far from that size. There is a positive correlation between the size of a predator and its prey. 28. A predator may assess a patch and decide whether to spend time searching for prey in it. 25. This may involve some knowledge of the preferences of the prey. For example, ladybirds can choose a patch of vegetation suitable for their aphid prey. 38. Capture. To capture prey, predators have a spectrum of pursuit modes that range from overt chase, pursuit predation, to a sudden strike on nearby prey, ambush predation. 25-39-12 Another strategy in between ambush and pursuit is ballistic interception, where a predator observes and predicts a prey's motion and then launches its attack accordingly. 40. Ambush. Western green lizard ambushes its grasshopper prey. A trapdoor spider waiting in its burrow to ambush its prey. Ambush or sit-and-wait predators are carnivorous animals that capture prey by stealth or surprise. In animals, ambush predation is characterized by the predator scanning the environment from a concealed position until a prey is spotted, and then rapidly executing a fixed surprise attack. 41-40 Vertebrate ambush predators include frogs, fish such as the angel shark, the northern pike and the eastern frogfish. 40-42-43-44 Among the many invertebrate ambush predators are trapdoor spiders and Australian crab spiders on land and mantis shrimps in the sea. 41-45-46 Ambush predators often construct a burrow in which to hide, improving concealment at the cost of reducing their field of vision. Some ambush predators also use lures to attract prey within striking range. 40 The capturing movement has to be rapid to trap the prey, given that the attack is not modifiable once launched. 40. Ballistic interception. The chameleon attacks prey by shooting out its tongue. Ballistic interception is the strategy where a predator observes the movement of a prey, predicts its motion, works out an interception path, and then attacks the prey on that path. This differs from ambush predation in that the predator adjusts its attack according to how the prey is moving. 40 Ballistic interception involves a brief period for planning, giving the prey an opportunity to escape. Some frogs wait until snakes have begun their strike before jumping, reducing the time available to the snake to recalibrate its attack, and maximizing the angular adjustment that the snake would need to make to intercept the frog in real time. 40 Ballistic predators include insects such as dragonflies, and vertebrates such as archerfish, attacking with a jet of water, chameleons, attacking with their tongues, and some colobrid snakes. 40. Pursuit. Humpback whales are lunge feeders, filtering thousands of krill from seawater and swallowing them alive. Dragonflies, like this common clubtail with captured prey, are invertebrate pursuit predators. In pursuit predation, predators chase fleeing prey. If the prey flees in a straight line, capture depends only on the predators being faster than the prey. 40 If the prey maneuvers by turning as it flees, the predator must react in real time to calculate and follow a new intercept path, such as by parallel navigation, as it closes on the prey. 40 Many pursuit predators use camouflage to approach the prey as close as possible unobserved, stalking before starting the pursuit. 40 Pursuit predators include terrestrial mammals such as humans, African wild dogs, spotted hyenas and wolves, marine predators such as dolphins, orcas and many predatory fishes, such as tuna. 47-48 Predatory birds, raptors, such as falcons, and insects such as dragonflies. 49. An extreme form of pursuit is endurance or persistence hunting, in which the predator tires out the prey by following it over a long distance, sometimes for hours at a time. The method is used by human hunter-gatherers and by canids such as African wild dogs and domestic hounds. The African wild dog is an extreme persistence predator, tiring out individual prey by following them for many miles at relatively low speed. 50. A specialized form of pursuit predation is the lunge feeding of baleen whales. These very large marine predators feed on plankton, especially krill, diving and actively swimming into concentrations of plankton, and then taking a huge gulp of water and filtering it through their feathery baleen. Plates. 51-52. Pursuit predators may be social, like the lion and wolf that hunt in groups, or solitary. 2. Handling. Catfish has sharp dorsal and pectoral spines which it holds erect to discourage predators such as herons which swallow prey whole. Osprey tears its fish prey apart, avoiding dangers such as sharp spines. Once the predator has captured the prey, it has to handle it very carefully if the prey is dangerous to eat, such as if it possesses sharp or poisonous spines, as in many prey fish. Some catfish such as the Ictaluridae have spines on the back dorsal and belly pectoral, which lock in the erect position. As the catfish thrashes about when captured, these could pierce the predator's mouth, possibly fatally. Some fish-eating birds like the osprey avoid the danger of spines by tearing up their prey before eating it. 53. Solitary versus social predation. In social predation, a group of predators cooperates to kill prey. This makes it possible to kill creatures larger than those they could overpower singly. For example, hyenas and wolves collaborate to catch and kill herbivores as large as buffalo, and lions even hunt elephants. 54, 55, 56. It can also make prey more readily available through strategies like flushing of prey and herding it into a smaller area. For example, when mixed flocks of birds forage, the birds in front flush out insects that are caught by the birds behind. Spinner dolphins form a circle around a school of fish and move inwards, concentrating the fish by a factor of 200. 57. By hunting socially, chimpanzees can catch colobus monkeys that would readily escape an individual hunter, while cooperating Harris hawks can trap rabbits. 54, 58. Wolves, social predators, cooperate to hunt and kill bison. Predators of different species sometimes cooperate to catch prey. In coral reefs, when fish such as the grouper and coral trout spot prey that is inaccessible to them, they signal to giant moray eels, Napoleon wrasses or octopuses. These predators are able to access small crevices and flush out the prey. 59, 60. Killer whales have been known to help whalers hunt baleen whales. 61. Social hunting allows predators to tackle a wider range of prey, but at the risk of competition for the captured food. Solitary predators have more chance of eating what they catch, at the price of increased expenditure of energy to catch it, and increased risk that the prey will escape. 62, 63. Ambush predators are often solitary to reduce the risk of becoming prey themselves. 64. Of 245 terrestrial members of the carnivora, the group that includes the cats, dogs, and bears, 177 are solitary, and 35 of the 37 wild cats are solitary, 65 including the cougar and cheetah. 62, 2. However, the solitary cougar does allow other cougars to share in a kill. 66. And the coyote can be either solitary or social. 67. Other solitary predators include the northern pike. 68. Wolf spiders and all the thousands of species of solitary wasps among arthropods. 69, 70. And many microorganisms in zooplankton. 22, 71. Specialization. Physical adaptations. Under the pressure of natural selection, predators have evolved a variety of physical adaptations for detecting, catching, killing, and digesting prey. These include speed, agility, stealth, sharp senses, claws, teeth, filters, and suitable digestive systems. 72. For detecting prey, predators have well-developed vision, smell, or hearing. 12. Predators as diverse as owls and jumping spiders have forward-facing eyes, providing accurate binocular vision over a relatively narrow field of view, whereas prey animals often have less acute all-round vision. Animals such as foxes can smell their prey even when it is concealed under 2 feet, 60 centimeters, of snow or earth. Many predators have acute hearing, and some such as echolocating bats hunt exclusively by active or passive use of sound. 73. Predators including big cats, birds of prey, and ants share powerful jaws, sharp teeth, or claws which they use to seize and kill their prey. Some predators such as snakes and fish-eating birds like herons and cormorants swallow their prey whole. Some snakes can unhinge their jaws to allow them to swallow large prey, while fish-eating birds have long spear-like beaks that they use to stab and grip fast-moving and slippery prey. 73. Fish and other predators have developed the ability to crush or open the armored shells of mollusks. 74. Many predators are powerfully built and can catch and kill animals larger than themselves. This applies as much to small predators such as ants and shrews as to big and visibly muscular carnivores like the cougar and lion. 73. 2. 75. Skull of brown bear has large pointed canines for killing prey, and self-sharpening carnassial teeth that rear for cutting flesh with a scissor-like action. Eyes, sensitive antennae, mandibles of jack jumper ant. Crab spider, an ambush predator with forward-facing eyes, catching another predator, a field digger wasp. Red-tailed hawk uses sharp hooked claws and beak to kill and tear up its prey specialist, a great blue heron with a speared fish. Indian python unhinges its jaw to swallow large prey like this chital. Diet and behavior. It is difficult to determine whether given adaptations are truly the result of coevolution, where a prey adaptation gives rise to a predator adaptation that is countered by further adaptation in the prey. An alternative explanation is escalation, where predators are adapting to competitors, their own predators are dangerous prey. 123. Apparent adaptations to predation may also have arisen for other reasons and then been co-opted for attack or defense. In some of the insects preyed on by bats, hearing evolved before bats appeared and was used to hear signals used for territorial defense and mating. 124. Their hearing evolved in response to bat predation, but the only clear example of reciprocal adaptation in bats is stealth echolocation. 125. A more symmetric arms race may occur when the prey are dangerous, having spines, quills, toxins or venom that can harm the predator. The predator can respond with avoidance, which in turn drives the evolution of mimicry. Avoidance is not necessarily an evolutionary response, as it is generally learned from bad experiences with prey. However, when the prey is capable of killing the predator, as can a coral snake with its venom, there is no opportunity for learning and avoidance must be inherited. Predators can also respond to dangerous prey with counter-adaptations. In western North America, the common garter snake has developed a resistance to the toxin in the skin of the rope-skin newt. 122. Role in ecosystems. Predators affect their ecosystems not only directly by eating their own prey, but by indirect means such as reducing predation by other species, or altering the foraging behavior of a herbivore, as with the biodiversity effect of wolves on riverside vegetation or sea otters on kelp forests. This may explain population dynamics effects such as the cycles observed in lynx and snowshoe. Hares. 126, 127, 128. Trophic level. One way of classifying predators is by trophic level. Carnivores that feed on herbivores are secondary consumers, their predators are tertiary consumers, and so forth. 129. At the top of this food chain are apex predators such as lions. 130. Many predators however eat from multiple levels of the food chain. A carnivore may eat both secondary and tertiary consumers. 131. This means that many predators must contend with intraguild predation, where other predators kill and eat them. For example, coyotes compete with and sometimes kill gray foxes and bobcats. 132. Trophic transfer. Trophic transfer within an ecosystem refers to the transport of energy and nutrients as a result of predation. Energy passes from one trophic level to the next as predators consume organic matter from another organism's body. Within each transfer, while there are uses of energy, there are also losses of energy. Marine trophic levels vary depending on locality and the size of the primary producers. There are generally up to 6 trophic levels in the open ocean, 4 over continental shelves, and around 3 in upwelling zones. 133. For example, a marine habitat with 5 trophic levels could be represented as follows. Herbivores feed primarily on phytoplankton, carnivores feed primarily on other zooplankton, animals detritivores feed primarily on dead organic matter, detritus. Omnivores feed on a mixed diet of phyto and zooplankton and detritus, and mixotrophs which combine autotrophy, using light energy to grow without intake of any additional organic compounds or nutrients, with heterotrophy, feeding on other plants and animals for energy and nutrients, herbivores, omnivores and carnivores, and detritivores. Trophic transfer efficiency measures how effectively energy is transferred or passed up through higher trophic levels of the marine food web. As energy moves up the trophic levels, it decreases due to heat, waste, and the natural metabolic processes that occur as predators consume their prey. The result is that only about 10% of the energy at any trophic level is transferred to the next level. This is often referred to as the 10% rule, which limits the number of trophic levels that an individual ecosystem is capable of supporting. 134. Biodiversity maintained by apex predation. Predators may increase the biodiversity of communities by preventing a single species from becoming dominant. Predators may increase the biodiversity of communities by preventing a single species from becoming dominant. Such predators are known as keystone species and may have a profound influence on the balance of organisms in a particular ecosystem. 135. Introduction or removal of this predator, or changes in its population density, can have drastic cascading effects on the equilibrium of many other populations in the ecosystem. For example, grazers of a grassland may prevent a single dominant species from taking over. 136. Riparian willow recovery at Blacktail Creek, Yellowstone National Park, after reintroduction of wolves, the local keystone species and apex predator. 137. Left, in 2002, right, in 2015. The elimination of wolves from Yellowstone National Park had profound impacts on the trophic pyramid. In that area, wolves are both keystone species and apex predators. Without predation, herbivores began to overgraze many woody browse species, affecting the area's plant populations. In addition, wolves often kept animals from grazing near streams, protecting the beaver's food sources. The removal of wolves had a direct effect on the beaver population, as their habitat became territory for grazing. Increased browsing on willows and conifers along Blacktail Creek due to a lack of predation caused channel incision because the reduced beaver population was no longer able to slow the water down and keep the soil in place. The predators were thus demonstrated to be of vital importance in the ecosystem. 137. Population dynamics. Numbers of snowshoe hare, Lepus americanus, yellow background, and Canada lynx, black line, foreground furs sold to the Hudson's Bay Company from 1845 to 1935. In the absence of predators, the population of a species can grow exponentially until it approaches the carrying capacity of the environment. 138. Predators limit the growth of prey both by consuming them and by changing their behavior. 139. Increases or decreases in the prey population can also lead to increases or decreases in the number of predators, for example, through an increase in the number of young they bear. Cyclical fluctuations have been seen in populations of predator and prey, often with offsets between the predator and prey cycles. A well-known example is that of the snowshoe hare and lynx. Over a broad span of boreal forests in Alaska and Canada, the hare populations fluctuate in near synchrony with a 10-year period, and the lynx populations fluctuate in response. This was first seen in historical records of animals caught by fur hunters for the Hudson's Bay Company over more than a century. 140, 128, 141, 142. Predator-prey population cycles in a Lotka-Volterra model. A simple model of a system with one species each of predator and prey, the Lotka-Volterra equations, predicts population cycles. 143. However, attempts to reproduce the predictions of this model in the laboratory have often failed. For example, when the protozoan Didinium nasutum is added to a culture containing its prey, Paramecium caudatum, the latter is often driven to extinction. 144. The Lotka-Volterra equations rely on several simplifying assumptions, and they are structurally unstable, meaning that any change in the equations can stabilize or destabilize the dynamics. 145. 146. For example, one assumption is that predators have a linear functional response to prey. The rate of kills increases in proportion to the rate of encounters. If this rate is limited by time spent handling each catch, then prey populations can reach densities above which predators cannot control them. 144. Another assumption is that all prey individuals are identical. In reality, predators tend to select young, weak, and ill individuals, leaving prey populations able to regrow. 147. Many factors can stabilize predator and prey populations. 148. One example is the presence of multiple predators, particularly generalists that are attracted to a given prey species if it is abundant and look elsewhere if it is not. 149. As a result, population cycles tend to be found in northern temperate and subarctic ecosystems because the food webs are simpler. 150. The snowshoe hare-lynx system is subarctic, but even this involves other predators, including coyotes, goshawks and great horned owls, and the cycle is reinforced by variations in the food available to the hares. 151. A range of mathematical models have been developed by relaxing the assumptions made in the Lotka-Volterra model. These variously allow animals to have geographic distributions, or to migrate, to have differences between individuals, such as sexes and an age structure, so that only some individuals reproduce, to live in a varying environment, such as with changing seasons. 152, 153, and analyzing the interactions of more than just two species at once. Such models predict widely differing and often chaotic predator-prey population dynamics. 152, 154. The presence of refuge areas, where prey are safe from predators, may enable prey to maintain larger populations but may also destabilize the dynamics. 155, 156, 157, 158. Evolutionary history. Predation dates from before the rise of commonly recognized carnivores by hundreds of millions, perhaps billions, of years. Predation has evolved repeatedly in different groups of organisms. 159. The rise of eukaryotic cells at around 2.7 Gaia, the rise of multicellular organisms at about 2 Gaia, and the rise of mobile predators around 600 Maya 2 Gaia, probably around 1 Gaia, have all been attributed to early predatory behavior, and many very early remains show evidence of boreholes or other markings attributed to small predator species. 5. It likely triggered major evolutionary transitions including the arrival of cells, eukaryotes, sexual reproduction, multicellularity, increased size, mobility including insect flight, 160, and armored shells and exoskeletons. 5. The earliest predators were microbial organisms, which engulfed or grazed on others. Because the fossil record is poor, these first predators could date back anywhere between 1 and over 2.7 Gaia billion years ago. 5. Predation visibly became important shortly before the Cambrian period, around 550 million years ago, as evidenced by the almost simultaneous development of calcification in animals and algae, 161, and predation avoiding burrowing. However, predators had been grazing on microorganisms since at least 1,000 million years ago. 162, 163, with evidence of selective rather than random predation from a similar time. 164. Auroralumina attenboroughi is an ediacaran crown group cnidarian, 557 to 562 Maya, some 20 million years before the Cambrian explosion, from Charnwood Forest, England. It is thought to be one of the earliest predatory animals, catching small prey with its nematocysts as modern cnidarians. 165. The fossil record demonstrates a long history of interactions between predators and their prey from the Cambrian period onwards, showing for example that some predators drilled through the shells of bivalve and gastropod mollusks, while others ate these organisms by breaking their shells. 166. Among the Cambrian predators were invertebrates like the anomalocaridids with appendages suitable for grabbing prey, large compound eyes and jaws made of a hard material like that in the exoskeleton of an insect. 167. Some of the first fish to have jaws were the armored and mainly predatory placoderms of the Silurian to Devonian periods, one of which, the 6 meters 20 feet Dunkleosteus, is considered the world's first vertebrate, superpredator, preying upon other predators. 168, 169. Insects developed the ability to fly in the early Carboniferous or late Devonian, enabling them among other things to escape from predators. 160. Among the largest predators that have ever lived were the theropod dinosaurs such as Tyrannosaurus from the Cretaceous period. They preyed upon herbivorous dinosaurs such as hadrosaurs, ceratopsians and ankylosaurs. 170. Auroralumina attenboroughi, an ediacaran predator, circa 560 Maya. It was a stem group cnidarian, catching prey with its nematocysts. 165. The Cambrian substrate revolution saw life on the seafloor change from minimal burrowing left to a diverse burrowing fauna right, probably to avoid new Cambrian predators. The anomalocaridae Peytoia, a Cambrian invertebrate, probably an apex predator. Meganoromani, a predatory carboniferous insect related to dragonflies, could fly to escape terrestrial predators. Its large size, with a wingspan of 65 centimeters 30 in, may reflect the lack of vertebrate aerial predators at that time. Tyrannosaurus, a large theropod dinosaur of the Cretaceous, reconstruction. Dunkleosteus terrelli, a Devonian placoderm, perhaps. The world's first vertebrate superpredator, reconstruction. In human society. Sand hunter, Botswana. Practical uses. Humans, as omnivores, are to some extent predatory. 171. Using weapons and tools to fish. 172. Hunt and trap animals. 173. They also use other predatory species such as dogs, cormorants, 174, and falcons to catch prey for food or for sport. 175. Two mid-sized predators, dogs and cats, are the animals most often kept as pets in Western societies. 176. 177. Human hunters, including the San of southern Africa, use persistence hunting, a form of pursuit predation where the pursuer may be slower than prey such as a kudu antelope over short distances, but follows it in the midday heat until it is exhausted, a pursuit that can take up to 5 hours. 178. 179. In biological pest control, predators and parasitoids from a pest's natural range are introduced to control populations, at the risk of causing unforeseen problems. Natural predators, provided they do no harm to non-pest species, are an environmentally friendly and sustainable way of reducing damage to crops and an alternative to the use of chemical agents such as pesticides. 180. Symbolic uses. The Capitoline wolf suckling Romulus and Remus, the mythical founders of Rome. In film, the idea of the predator as a dangerous if humanoid enemy is used in the 1987 science fiction horror action film Predator and its 3 sequels. 181. 182. A terrifying predator, a gigantic man-eater, man-eating great white shark, is central, too, to Steven Spielberg's 1974 thriller Jaws. 183. Among poetry on the theme of predation, a predator's consciousness might be explored, such as in Ted Hughes's Pike. 184. The phrase, nature, red in tooth and claw, from Alfred, Lord Tennyson's 1849 poem, In Memoriam Ahh. Has been interpreted as referring to the struggle between predators and prey. 185. In mythology and folk fable, predators such as the fox and wolf have mixed reputations. 186. The fox was a symbol of fertility in ancient Greece, but a weather demon in northern Europe, and a creature of the devil in early Christianity. The fox is presented as sly, greedy, and cunning in fables from Aesop onwards. 186. The big bad wolf is known to children in tales such as Little Red Riding Hood, but is a demonic figure in the Icelandic Edda sagas, where the wolf Fenrir appears in the apocalyptic ending of the world. 186. In the Middle Ages, belief spread in werewolves, men transformed into wolves. 186. In ancient Rome, and in ancient Egypt, the wolf was worshipped, the she-wolf appearing in the founding myth of Rome, suckling Romulus and Remus. 186. More recently, in Rudyard Kipling's 1894 The Jungle Book, Mowgli is raised by the wolf pack. 186. Attitudes to large predators in North America, such as wolf, grizzly bear and cougar, have shifted from hostility or ambivalence, accompanied by active persecution, towards positive and protective in the second half of the 20th century. Herbivore. A herbivore is an animal anatomically and physiologically evolved to feed on plants, especially upon vascular tissues such as foliage, fruits or seeds, as the main component of its diet. These more broadly also encompass animals that eat non-vascular autotrophs such as mosses, algae and lichens, but do not include those feeding on decomposed plant matters, i.e. detritivores, or macrofungi, i.e. fungivores. A deer and 2 fawns feeding on foliage. A sawfly larva feeding on a leaf. Tracks made by terrestrial gastropods with their radullas, scraping green algae from a surface inside a greenhouse. As a result of their plant-based diet, herbivorous animals typically have mouth structures, jaws or mouthparts well adapted to mechanically break down plant materials, and their digestive systems have special enzymes, e.g. amylase and cellulase, to digest polysaccharides. Grazing herbivores such as horses and cattle have wide flat crown teeth that are better adapted for grinding grass, tree bark and other tougher lignin-containing materials, and many of them evolved rumination or cecotropic behaviors to better extract nutrients from plants. A large percentage of herbivores also have mutualistic gut flora made up of bacteria and protozoans that help to degrade the cellulose in plants, one whose heavily cross-linking polymer structure makes it far more difficult to digest than the protein and fat-rich animal tissues that carnivores eat. 2. Etymology. Herbivore is the anglicized form of a modern Latin coinage, herbivora, cited in Charles Lyell's 1830 Principles of Geology. 3. Richard Owen employed the anglicized term in an 1854 work on fossil teeth and skeletons. 3. Herbivora is derived from Latin herba small plant, herb, 4. And vora, from vorare to eat, devour. 5. Definition and related terms. Herbivora is a form of consumption in which an organism principally eats autotrophs, 6, such as plants, algae and photosynthesizing bacteria. Herbivora is a form of consumption in which an organism principally eats autotrophs, 6, such as plants, algae and photosynthesizing bacteria. More generally, organisms that feed on autotrophs in general are known as primary consumers. Herbivora is usually limited to animals that eat plants. Insect herbivora can cause a variety of physical and metabolic alterations in the way the host plant interacts with itself and other surrounding biotic factors. 7. 8. Fungi, bacteria, and protists that feed on living plants are usually termed plant pathogens, plant diseases, while fungi and microbes that feed on dead plants are described as saprotrophs. Flowering plants that obtain nutrition from other living plants are usually termed parasitic plants. There is, however, no single exclusive and definitive ecological classification of consumption patterns. Each textbook has its own variations on the Theme 9, 10, 11. Evolution of herbivora. A fossil viburnum lesqueroic sea leaf with evidence of insect herbivora, Dakota Sandstone Cretaceous of Ellsworth County, Kansas. Scale bar is 10 mm. The understanding of herbivora in geological time comes from three sources: fossilized plants, which may preserve evidence of defense, such as spines or herbivora-related damage, the observation of plant debris and fossilized animal feces, and the construction of herbivore mouthparts. 12. Although herbivora was long thought to be a Mesozoic phenomenon, fossils have shown that plants were being consumed by arthropods within less than 20 million years after the first land plants evolved. 13. Insects fed on the spores of early Devonian plants, and the Rhynie chert also provides evidence that organisms fed on plants using a Pearson-suck technique. 12. During the next 75 million years, plants evolved a range of more complex organs, such as roots and seeds. There is no evidence of any organism being fed upon until the Middle-Late Mississippian, 330.9 million years ago. There was a gap of 50 to 100 million years between the time each organ evolved and the time organisms evolved to feed upon them. This may be due to the low levels of oxygen during this period, which may have suppressed evolution. 13. Further than their arthropod status, the identity of these early herbivores is uncertain. 13. Pole feeding and skeletonization are recorded in the early Permian, with surface fluid feeding evolving by the end of that period. 12. Herbivora among four-limbed terrestrial vertebrates, the tetrapods, developed in the late Carboniferous, 307 to 99 million years ago. 14. The oldest known example being Desmodon hesperus. 15. Early tetrapods were large amphibious piscivores. While amphibians continued to feed on fish and insects, some reptiles began exploring two new food types: tetrapods carnivory and plants herbivora. The entire dinosaur order Ornithischia was composed of herbivorous dinosaurs. 14. Carnivory was a natural transition from insectivory for medium and large tetrapods, requiring minimal adaptation. In contrast, a complex set of adaptations was necessary for feeding on highly fibrous plant materials. 14. Arthropods evolved herbivora in four phases, changing their approach to it in response to changing plant communities. 16. Tetrapod herbivores made their first appearance in the fossil record of their jaws near the Permio-Carboniferous boundary, approximately 300 million years ago. The earliest evidence of the herbivora has been attributed to dental occlusion, the process in which teeth from the upper jaw come in contact with teeth in the lower jaw is present. The evolution of dental occlusion led to a drastic increase in plant food processing and provides evidence about feeding strategies based on tooth wear patterns. Examination of phylogenetic frameworks of tooth and jaw morphologies has revealed that dental occlusion developed independently in several lineages tetrapod herbivores. This suggests that evolution and spread occurred simultaneously within various lineages. 17. Food chain. Leaf miners feed on leaf tissue between the epidermal layers, leaving visible trails. Herbivores form an important link in the food chain because they consume plants to digest the carbohydrates photosynthetically produced by a plant. Carnivores in turn consume herbivores for the same reason, while omnivores can obtain their nutrients from either plants or animals. Due to a herbivore's ability to survive solely on tough and fibrous plant matter, they are termed the primary consumers in the food cycle chain. Herbivora, carnivory, and omnivory can be regarded as special cases of consumer-resource interactions. 18. Feeding strategies. Two herbivore feeding strategies are grazing, e.g. cows, and browsing, e.g. moose. For a terrestrial mammal to be called a grazer, at least 90% of the forage has to be grass, and for a browser at least 90% tree leaves and twigs. An intermediate feeding strategy is called mixed feeding. 19. In their daily need to take up energy from forage, herbivores of different body mass may be selective in choosing their food. 20. Selective means that herbivores may choose their forage source depending on, e.g., season or food availability, but also that they may choose high quality and consequently highly nutritious forage before lower quality. The latter especially is determined by the body mass of the herbivore, with small herbivores selecting for high quality forage, and with increasing body mass animals are less selective. 20. Several theories attempt to explain and quantify the relationship between animals and their food, such as Kleiber's law, Holling's disk equation and the marginal value theorem, see below. Kleiber's law describes the relationship between an animal's size and its feeding strategy, saying that larger animals need to eat less food per unit weight than smaller animals. 21. Kleiber's law states that the metabolic rate Q0 of an animal is the mass of the animal m raised to the 3/4 power, Q0 equals m 3/4. Therefore, the mass of the animal increases at a faster rate than the metabolic rate. 21. Herbivores employ numerous types of feeding strategies. Many herbivores do not fall into one specific feeding strategy, but employ several strategies and eat a variety of plant parts. Types of feeding strategies. Feeding strategy diet examples. Algivores: algae, krill, crabs, sea snail, sea urchin, parrotfish, surgeonfish, flamingo. Frugivores: fruit, rough lemurs, orangutans. Folivores: leaves, koalas, gorillas, red colubuses, many leaf beetles. Nectarivores: nectar, honey possums, hummingbirds. Granivores: seeds, Hawaiian honeycreepers, bean weevils. Graminivores: grass horses. Pollinivores: pollen bees. Muscivores: plant fluids, i.e. sap aphids. Xylophages: wood termites, longicorn beetles, ambrosia beetles. Optimal foraging theory is a model for predicting animal behavior while looking for food or other resources, such as shelter or water. This model assesses both individual movement, such as animal behavior while looking for food, and distribution within a habitat, such as dynamics at the population and community level. For example, the model would be used to look at the browsing behavior of a deer while looking for food, as well as that deer's specific location and movement within the forested habitat and its interaction with other deer while in that habitat. 22. This model has been criticized as circular and untestable. Critics have pointed out that its proponents use examples that fit the theory, but do not use the model when it does not fit the reality. 23-24 Other critics point out that animals do not have the ability to assess and maximize their potential gains, therefore the optimal foraging theory is irrelevant and derived to explain trends that do not exist in nature. 25-26 Holling's disk equation models the efficiency at which predators consume prey. The model predicts that as the number of prey increases, the amount of time predators spend handling prey also increases, and therefore the efficiency of the predator decreases. 27 In 1959, S. Holling proposed an equation to model the rate of return for an optimal diet. Rate R equals energy gained in foraging EF time searching Ts plus time handling TH. Where S equals cost of search per unit time, F equals rate of encounter with items, H equals handling time, E equals energy gain per encounter. In effect, this would indicate that a herbivore in a dense forest would spend more time handling, eating, the vegetation because there was so much vegetation around than a herbivore in a sparse forest, who could easily browse through the forest vegetation. According to the Holling's disk equation, a herbivore in the sparse forest would be more efficient at eating than the herbivore in the dense forest. The marginal value theorem describes the balance between eating all the food in a patch for immediate energy, or moving to a new patch and leaving the plants in the first patch to regenerate for future use. The theory predicts that absent complicating factors, an animal should leave a resource patch when the rate of payoff amount of food falls below the average rate of payoff for the entire area. 28 According to this theory, an animal should move to a new patch of food when the patch they are currently feeding on requires more energy to obtain food than an average patch. Within this theory, two subsequent parameters emerge, the giving up density GUD and the giving up time GUT. The giving up density GUD quantifies the amount of food that remains in a patch when a forager moves to a new patch. 29 The giving up time GUT is used when an animal continuously assesses the patch quality. 30. Plant-herbivore interactions. Interactions between plants and herbivores can play a prevalent role in ecosystem dynamics such as community structure and functional processes. 31-32 Plant diversity and distribution is often driven by herbivora, and it is likely that trade-offs between plant competitiveness and defensiveness, and between colonization and mortality allow for coexistence between species in the presence of herbivores. 33, 34, 35, 36 However, the effects of herbivora on plant diversity and richness is variable. For example, increased abundance of herbivores such as deer decrease plant diversity and species richness. 37 While other large mammalian herbivores like bison control dominant species which allows other species to flourish. 38 Plant-herbivore interactions can also operate so that plant communities mediate herbivore communities. 39 Plant communities that are more diverse typically sustain greater herbivore richness by providing a greater and more diverse set of resources. 40 Coevolution and phylogenetic correlation between herbivores and plants are important aspects of the influence of herbivore and plant interactions on communities and ecosystem functioning, especially in regard to herbivorous insects. 32, 39, 41 This is apparent in the adaptations plants develop to tolerate and/or defend from insect herbivora and the responses of herbivores to overcome these adaptations. The evolution of antagonistic and mutualistic plant-herbivore interactions are not mutually exclusive and may co-occur. 42 Plant phylogeny has been found to facilitate the colonization and community assembly of herbivores, and there is evidence of phylogenetic linkage between plant beta diversity and phylogenetic beta diversity of insect clades such as butterflies. 39 These types of eco-evolutionary feedbacks between plants and herbivores are likely the main driving force behind plant and herbivore diversity. 39, 43 Abiotic factors such as climate and biogeographical features also impact plant-herbivore communities and interactions. For example, in temperate freshwater wetlands herbivorous waterfowl communities change according to season, with species that eat above-ground vegetation being abundant during summer, and species that forage below ground being present in winter months. 31, 36 These seasonal herbivore communities differ in both their assemblage and functions within the wetland ecosystem. 36 Such differences in herbivore modalities can potentially lead to trade-offs that influence species traits and may lead to additive effects on community composition and ecosystem functioning. 31, 36 Seasonal changes and environmental gradients such as elevation and latitude often affect the palatability of plants which in turn influences herbivore community assemblages and vice versa. 32, 44 Examples include a decrease in abundance of leaf-chewing larvae in the fall when hardwood leaf palatability decreases due to increased tannin levels which results in a decline of arthropod species richness. 45 And increased palatability of plant communities at higher elevations where grasshoppers' abundances are lower. 32 Climatic stressors such as ocean acidification can lead to responses in plant-herbivore interactions in relation to palatability as well. 46 Herbivore offense. Aphids are fluid feeders on plant sap. The myriad defenses displayed by plants means that their herbivores need a variety of skills to overcome these defenses and obtain food. These allow herbivores to increase their feeding and use of a host plant. Herbivores have three primary strategies for dealing with plant defenses: choice, herbivore modification, and plant modification. Feeding choice involves which plants a herbivore chooses to consume. It has been suggested that many herbivores feed on a variety of plants to balance their nutrient uptake and to avoid consuming too much of any one type of defensive chemical. This involves a trade-off, however, between foraging on many plant species to avoid toxins or specializing on one type of plant that can be detoxified. 47. Herbivore modification is when various adaptations to body or digestive systems of the herbivore allow them to overcome plant defenses. This might include detoxifying secondary metabolites, 48. Sequestering toxins unaltered, 49. Or avoiding toxins, such as through the production of large amounts of saliva to reduce effectiveness of defenses. Herbivores may also utilize symbionts to evade plant defenses. For example, some aphids use bacteria in their gut to provide essential amino acids lacking in their sap diet. 50. Plant modification occurs when herbivores manipulate their plant prey to increase feeding. For example, some caterpillars roll leaves to reduce the effectiveness of plant defenses activated by sunlight. 51. Plant defense. A plant defense is a trait that increases plant fitness when faced with herbivora. This is measured relative to another plant that lacks the defensive trait. Plant defenses increase survival and/or reproduction fitness of plants under pressure of predation from herbivores. Defense can be divided into two main categories, tolerance and resistance. Tolerance is the ability of a plant to withstand damage without a reduction in fitness. 52. This can occur by diverting herbivora to non-essential plant parts, resource allocation, compensatory growth, or by rapid regrowth and recovery from herbivora. 53. Resistance refers to the ability of a plant to reduce the amount of damage it receives from herbivores. 52. This can occur via avoidance in space or time. 54. Physical defenses, or chemical defenses. Defenses can either be constitutive, always present in the plant, or induced, produced or translocated by the plant following damage or stress. 55. Physical, or mechanical, defenses are barriers or structures designed to deter herbivores or reduce intake rates, lowering overall herbivora. Thorns such as those found on roses or acacia trees are one example, as are the spines on a cactus. Smaller hairs known as trichomes may cover leaves or stems and are especially effective against invertebrate herbivores. 56. In addition, some plants have waxes or resins that alter their texture, making them difficult to eat. Also, the incorporation of silica into cell walls is analogous to that of the role of lignin in that it is a compression-resistant structural component of cell walls, so that plants with their cell walls impregnated with silica are thereby afforded a measure of protection against herbivora. 57. Chemical defenses are secondary metabolites produced by the plant that deter herbivora. There are a wide variety of these in nature, and a single plant can have hundreds of different chemical defenses. Chemical defenses can be divided into two main groups, carbon-based defenses and nitrogen-based defenses. 58. 1. Carbon-based defenses include terpenes and phenolics. Terpenes are derived from 5-carbon isoprene units and comprise essential oils, carotenoids, resins, and latex. They can have several functions that disrupt herbivores, such as inhibiting adenosine triphosphate, ATP, formation, molting hormones, or the nervous system. 59. Phenolics combine an aromatic carbon ring with a hydroxyl group. There are several different phenolics such as lignins, which are found in cell walls and are very indigestible except for specialized microorganisms, tannins, which have a bitter taste and bind to proteins making them indigestible, and furanocoumarins, which produce free radicals disrupting DNA, protein, and lipids, and can cause skin irritation. 2. Nitrogen-based defenses are synthesized from amino acids and primarily come in the form of alkaloids and cyanogens. Alkaloids include commonly recognized substances such as caffeine, nicotine, and morphine. These compounds are often bitter and can inhibit DNA or RNA synthesis or block nervous system signal transmission. Cyanogens get their name from the cyanide stored within their tissues. This is released when the plant is damaged and inhibits cellular respiration and electron transport. Plants have also changed features that enhance the probability of attracting natural enemies to herbivores. Some emit semiochemicals, odors that attract natural enemies, while others provide food and housing to maintain the natural enemy's presence, e.g. ants that reduce herbivora. 60. A given plant species often has many types of defensive mechanisms, mechanical or chemical, constitutive or induced, which allow it to escape from herbivores. 61. Predator-prey theory. According to the theory of predator-prey interactions, the relationship between herbivores and plants is cyclic. 62. When prey plants are numerous, their predators herbivores increase in numbers, reducing the prey population, which in turn causes predator number to decline. 62. The prey population eventually recovers, starting a new cycle. This suggests that the population of the herbivore fluctuates around the carrying capacity of the food source, in this case, the plant. Several factors play into these fluctuating populations and help stabilize predator-prey dynamics. For example, spatial heterogeneity is maintained, which means there will always be pockets of plants not found by herbivores. This stabilizing dynamic plays an especially important role for specialist herbivores that feed on one species of plant and prevents these specialists from wiping out their food source. 63. Prey defenses also help stabilize predator-prey dynamics, and for more information on these relationships see the section on plant defenses. Eating a second prey type helps herbivores' populations stabilize. 63. Alternating between two or more plant types provides population stability for the herbivore, while the populations of the plants oscillate. 62. This plays an important role for generalist herbivores that eat a variety of plants. Keystone herbivores keep vegetation populations in check and allow for a greater diversity of both herbivores and plants. 63. When an invasive herbivore or plant enters the system, the balance is thrown off and the diversity can collapse to a monotaxon system. 63. The back-and-forth relationship of plant defense and herbivore offense drives coevolution between plants and herbivores, resulting in a coevolutionary arms race. 48. 64. The escape and radiation mechanisms for coevolution presents the idea that adaptations in herbivores and their host plants has been the driving force behind speciation. 65. 66. Mutualism. While much of the interaction of herbivora and plant defense is negative, with one individual reducing the fitness of the other, some is beneficial. This beneficial herbivora takes the form of mutualisms in which both partners benefit in some way from the interaction. Seed dispersal by herbivores and pollination are two forms of mutualistic herbivora in which the herbivore receives a food resource and the plant is aided in reproduction. 67. Plants can also be indirectly affected by herbivores through nutrient recycling, with plants benefiting from herbivores when nutrients are recycled very efficiently. 42. Another form of plant-herbivore mutualism is physical changes to the environment and/or plant community structure by herbivores which serve as ecosystem engineers, such as wallowing by bison. 68. Swans form a mutual relationship with the plant species that they forage by digging and disturbing the sediment which removes competing plants and subsequently allows colonization of other plant species. 31. 36. Impacts. Mixed feeding shoal of herbivorous fish on a coral reef. Trophic cascades and environmental degradation. When herbivores are affected by trophic cascades, plant communities can be indirectly affected. 69. Often these effects are felt when predator populations decline and herbivore populations are no longer limited, which leads to intense herbivore foraging which can suppress plant communities. 70. With the size of herbivores having an effect on the amount of energy intake that is needed, larger herbivores need to forage on higher quality or more plants to gain the optimal amount of nutrients and energy compared to smaller herbivores. 71. Environmental degradation from white-tailed deer Odocoileus virginianus in the U.S. alone has the potential to both change vegetative communities. 72. Through overbrowsing and cost forest restoration projects upwards of $750 million annually. Another example of a trophic cascade involved plant-herbivore interactions are coral reef ecosystems. Herbivorous fish and marine animals are important algae and seaweed grazers, and in the absence of plant-eating fish, corals are outcompeted and seaweeds deprive corals of sunlight. 73. Economic impacts. Agricultural crop damage by the same species totals approximately $100 million every year. Insect crop damages also contribute largely to annual crop losses in the U.S. 74. Herbivores also affect economics through the revenue generated by hunting and ecotourism. For example, the hunting of herbivorous game species such as white-tailed deer, cottontail rabbits, antelope, and elk in the U.S. contributes greatly to the billion-dollar annually hunting industry. Ecotourism is a major source of revenue, particularly in Africa, where many large mammalian herbivores such as elephants, zebras, and giraffes help to bring in the equivalent of millions of U.S. dollars to various nations annually. Ecophysiology. Ecophysiology, from Greek oikos, household physis, nature, origin, and logia, environmental physiology or physiological ecology is a biological discipline that studies the response of an organism's physiology to environmental conditions. It is closely related to comparative physiology and evolutionary physiology. Ernst Haeckel's coinage bionomy is sometimes employed as a synonym. 1. Plants. Plant ecophysiology is concerned largely with two topics: mechanisms, how plants sense and respond to environmental change, and scaling or integration, how the responses to highly variable conditions, for example, gradients from full sunlight to 95% shade within tree canopies, are coordinated with one another, and how their collective effect on plant growth and gas exchange can be understood on this basis. In many cases, animals are able to escape unfavorable and changing environmental factors such as heat, cold, drought or floods, while plants are unable to move away and therefore must endure the adverse conditions or perish. Animals go places, plants grow places. Plants are therefore phenotypically plastic and have an impressive array of genes that aid in acclimating to changing conditions. It is hypothesized that this large number of genes can be partly explained by plant species' need to live in a wider range of conditions. Light. Light is the food of plants, i.e. the form of energy that plants use to build themselves and reproduce. The organs harvesting light in plants are leaves, and the process through which light is converted into biomass is photosynthesis. The response of photosynthesis to light is called light response curve of net photosynthesis (PI curve). The shape is typically described by a non-rectangular hyperbola. Three quantities of the light response curve are particularly useful in characterizing a plant's response to light intensities. The inclined asymptote has a positive slope representing the efficiency of light use, and is called quantum efficiency. The x-intercept is the light intensity at which biochemical assimilation, gross assimilation, balances leaf respiration so that the net CO2 exchange of the leaf is zero, called light compensation point, and a horizontal asymptote representing the maximum assimilation rate. Sometimes after reaching the maximum assimilation declines for processes collectively known as photoinhibition. 2. As with most abiotic factors, light intensity or radiance can be both suboptimal and excessive. Suboptimal light shade typically occurs at the base of a plant canopy or in an understory environment. Shade-tolerant plants have a range of adaptations to help them survive the altered quantity and quality of light typical of shade environments. Excess light occurs at the top of canopies and on open ground when cloud cover is low and the sun's zenith angle is low. Typically this occurs in the tropics and at high altitudes. Excess light incident on a leaf can result in photoinhibition and photodestruction. Plants adapted to high light environments have a range of adaptations to avoid or dissipate the excess light energy, as well as mechanisms that reduce the amount of injury caused. 3. Light intensity is also an important component in determining the temperature of plant organs' energy budget. 4. Temperature. In response to extremes of temperature, plants can produce various proteins. These protect them from the damaging effects of ice formation and falling rates of enzyme catalysis at low temperatures, and from enzyme denaturation and increased photorespiration at high temperatures. Ecophysiology. Ecophysiology, from Greek oikos, oikos, household physis, nature, origin, and logia, environmental physiology or physiological ecology is a biological discipline that studies the response of an organism's physiology to environmental conditions. It is closely related to comparative physiology and evolutionary physiology. Ernst Haeckel's coinage bionomy is sometimes employed as a synonym. 1. Plants. Plant ecophysiology is concerned largely with two topics: mechanisms, how plants sense and respond to environmental change, and scaling or integration, how the responses to highly variable conditions, for example, gradients from full sunlight to 95% shade within tree canopies, are coordinated with one another, and how their collective effect on plant growth and gas exchange can be understood on this basis. In many cases, animals are able to escape unfavorable and changing environmental factors such as heat, cold, drought or floods, while plants are unable to move away and therefore must endure the adverse conditions or perish. Animals go places, plants grow places. Plants are therefore phenotypically plastic and have an impressive array of genes that aid in acclimating to changing conditions. It is hypothesized that this large number of genes can be partly explained by plant species' need to live in a wider range of conditions. Light. Light is the food of plants, i.e. the form of energy that plants use to build themselves and reproduce. The organs harvesting light in plants are leaves, and the process through which light is converted into biomass is photosynthesis. The response of photosynthesis to light is called light response curve of net photosynthesis (PI curve). The shape is typically described by a non-rectangular hyperbola. Three quantities of the light response curve are particularly useful in characterizing a plant's response to light intensities. The inclined asymptote has a positive slope representing the efficiency of light use, and is called quantum efficiency. The x-intercept is the light intensity at which biochemical assimilation, gross assimilation, balances leaf respiration so that the net CO2 exchange of the leaf is zero, called light compensation point, and a horizontal asymptote representing the maximum assimilation rate. Sometimes after reaching the maximum, assimilation declines for processes collectively known as photoinhibition. 2. As with most abiotic factors, light intensity, or radiance, can be both suboptimal and excessive. Suboptimal light shade typically occurs at the base of a plant canopy or in an understory environment. Shade-tolerant plants have a range of adaptations to help them survive the altered quantity and quality of light typical of shade environments. Excess light occurs at the top of canopies and on open ground when cloud cover is low and the sun's zenith angle is low. Typically this occurs in the tropics and at high altitudes. Excess light incident on a leaf can result in photoinhibition and photodestruction. Plants adapted to high light environments have a range of adaptations to avoid or dissipate the excess light energy, as well as mechanisms that reduce the amount of injury caused. 3. Light intensity is also an important component in determining the temperature of plant organs' energy budget. 4. Temperature. In response to extremes of temperature, plants can produce various proteins. These protect them from the damaging effects of ice formation and falling rates of enzyme catalysis at low temperatures, and from enzyme denaturation and increased photorespiration at high temperatures. As temperatures fall, production of antifreeze proteins and dehydrins increases. As temperatures rise, production of heat shock proteins increases. Metabolic imbalances associated with temperature extremes result in the buildup of reactive oxygen species, which can be countered by antioxidant systems. Cell membranes are also affected by changes in temperature and can cause the membrane to lose its fluid properties and become a gel in cold conditions or to become leaky in hot conditions. This can affect the movement of compounds across the membrane. To prevent these changes, plants can change the composition of their membranes. In cold conditions, more unsaturated fatty acids are placed in the membrane, and in hot conditions, more saturated fatty acids are inserted. Infrared image showing the importance of transpiration in keeping leaves cool. Plants can avoid overheating by minimizing the amount of sunlight absorbed and by enhancing the cooling effects of wind and transpiration. Plants can reduce light absorption using reflective leaf hairs, scales, and waxes. These features are so common in warm, dry regions that these habitats can be seen to form a silvery landscape, as the light scatters off the canopies. 5. Some species, such as Macrophyllum purpureum, can move their leaves throughout the day so that they are always orientated to avoid the sun. Paraheliotropism. 6. Knowledge of these mechanisms has been key to breeding for heat stress tolerance in agricultural plants. Plants can avoid the full impact of low temperatures by altering their microclimate. For example, Rauwollia plants found in the uplands of New Zealand are said to resemble vegetable sheet, as they form tight cushion-like clumps to insulate the most vulnerable plant parts and shield them from cooling winds. The same principle has been applied in agriculture by using plastic mulch to insulate the growing points of crops in cool climates in order to boost plant growth. 7. Water. Too much or too little water can damage plants. If there is too little water, then tissues will dehydrate and the plant may die. If the soil becomes waterlogged, then the soil will become anoxic, low in oxygen, which can kill the roots of the plant. 8. The ability of plants to access water depends on the structure of their roots and on the water potential of the root cells. When soil water content is low, plants can alter their water potential to maintain a flow of water into the roots and up to the leaves, soil-plant atmosphere continuum. This remarkable mechanism allows plants to lift water as high as 120 meters by harnessing the gradient created by transpiration from the leaves. 9. In very dry soil, plants close their stomata to reduce transpiration and prevent water loss. The closing of the stomata is often mediated by chemical signals from the root, i.e., abscisic acid. In irrigated fields, the fact that plants close their stomata in response to drying of the roots can be exploited to trick plants into using less water without reducing yields. See partial root zone drying. The use of this technique was largely developed by Dr. Peter Dry and colleagues in Australia. 10. If drought continues, the plant tissues will dehydrate, resulting in a loss of turgor pressure that is visible as wilting. As well as closing their stomata, most plants can also respond to drought by altering their water potential, osmotic adjustment, and increasing root growth. Plants that are adapted to dry environments, xerophytes, have a range of more specialized mechanisms to maintain water and/or protect tissues when desiccation occurs. Waterlogging reduces the supply of oxygen to the roots and can kill a plant within days. Plants cannot avoid waterlogging, but many species overcome the lack of oxygen in the soil by transporting oxygen to the root from tissues that are not submerged. Species that are tolerant of waterlogging develop specialized roots near the soil surface and parenchyma to allow the diffusion of oxygen from the shoot to the root. Roots that are not killed outright may also switch to less oxygen-hungry forms of cellular respiration. 11. Species that are frequently submerged have evolved more elaborate mechanisms that maintain root oxygen levels, such as the aerial roots seen in mangrove forests. 12. However, for many terminally overwatered houseplants, the initial symptoms of waterlogging can resemble those due to drought. This is particularly true for flood-sensitive plants that show drooping of their leaves due to epinesty rather than wilting. CO2 concentration. CO2 is vital for plant growth, as it is the substrate for photosynthesis. Plants take in CO2 through stomatal pores on their leaves. At the same time as CO2 enters the stomata, moisture escapes. This trade-off between CO2 gain and water loss is central to plant productivity. The trade-off is all the more critical as rubisco, the enzyme used to capture CO2, is efficient only when there is a high concentration of CO2 in the leaf. Some plants overcome this difficulty by concentrating CO2 within their leaves using C4 carbon fixation or crassulation acid metabolism. However, most species use C3 carbon fixation and must open their stomata to take in CO2 whenever photosynthesis is taking place. Plant productivity in a warming world. The concentration of CO2 in the atmosphere is rising due to deforestation and the combustion of fossil fuels. This would be expected to increase the efficiency of photosynthesis and possibly increase the overall rate of plant growth. This possibility has attracted considerable interest in recent years, as an increased rate of plant growth could absorb some of the excess CO2 and reduce the rate of global warming. Extensive experiments growing plants under elevated CO2 using free air concentration enrichment have shown that photosynthetic efficiency does indeed increase. Plant growth rates also increase, by an average of 17% for above-ground tissue and 30% for below-ground tissue. 14. However, detrimental impacts of global warming, such as increased instances of heat and drought stress, mean that the overall effect is likely to be a reduction in plant productivity. 17. Reduced plant productivity would be expected to accelerate the rate of global warming. Overall, these observations point to the importance of avoiding further increases in atmospheric CO2 rather than risking runaway climate change. 18. Wind. Wind has 3 very different effects on plants. 19. It affects the exchanges of mass, water evaporation, CO2, and of energy, heat, between the plant and the atmosphere by renewing the air at the contact with the leaves, convection. It is sensed as a signal driving a wind acclimation syndrome by the plant known as thigmomorphogenesis, leading to modified growth and development and eventually to wind hardening. Its drag force can damage the plant, leaf abrasion, wind ruptures in branches and stems and wind throws and toppling in trees and lodging in crops. 20. Exchange of mass and energy. Wind influences the way leaves regulate moisture, heat, and carbon dioxide. When no wind is present, a layer of still air builds up around each leaf. This is known as the boundary layer and in effect insulates the leaf from the environment, providing an atmosphere rich in moisture and less prone to convective heating or cooling. As wind speed increases, the leaf environment becomes more closely linked to the surrounding environment. It may become difficult for the plant to retain moisture as it is exposed to dry air. On the other hand, a moderately high wind allows the plant to cool its leaves more easily when exposed to full sunlight. Plants are not entirely passive in their interaction with wind. Plants can make their leaves less vulnerable to changes in wind speed, by coating their leaves in fine hairs, trichomes, to break up the airflow and increase the boundary layer. In fact, leaf and canopy dimensions are often finely controlled to manipulate the boundary layer depending on the prevailing environmental conditions. 21. Acclimation. Plants can sense the wind through the deformation of its tissues. This signal leads to inhibits the elongation and stimulates the radial expansion of their shoots, while increasing the development of their root system. This syndrome of response is known as thigmomorphogenesis, results in shorter, stockier plants with strengthened stems, as well as to an improved anchorage. 22. It was once believed that this occurs mostly in very windy areas. But it has been found that it happens even in areas with moderate winds, so that wind-induced signal were found to be a major ecological factor. 23. Trees have a particularly well-developed capacity to reinforce their trunks when exposed to wind. From the practical side, this realization prompted arboriculturalists in the UK in the 1960s to move away from the practice of staking young amenity trees to offer artificial support. 24. Wind damage. Wind can damage most of the organs of the plants. Leaf abrasion due to the rubbing of leaves and branches or to the effect of airborne particles such as sand, and leaf of branch breakage are rather common phenomena that plants have to accommodate. In the more extreme cases, plants can be mortally damaged or uprooted by wind. This has been a major selective pressure acting over terrestrial plants. 25. Nowadays, it is one of the major threatening for agriculture and forestry even in temperate zones. 19. It is worse for agriculture in hurricane-prone regions, such as the banana-growing Windward Islands in the Caribbean. 26. When this type of disturbance occurs in natural systems, the only solution is to ensure that there is an adequate stock of seeds or seedlings to quickly take the place of the mature plants that have been lost. Although, in many cases, a successional stage will be needed before the ecosystem can be restored to its former state. Animals. Humans. The environment can have major influences on human physiology. Environmental effects on human physiology are numerous. One of the most carefully studied effects is the alterations in thermoregulation in the body due to outside stresses. This is necessary because in order for enzymes to function, blood to flow, and for various body organs to operate, temperature must remain at consistent, balanced levels. Thermoregulation. To achieve this, the body alters three main things to achieve a constant, normal body temperature: heat transfer to the epidermis, the rate of evaporation, the rate of heat production. The hypothalamus plays an important role in thermoregulation. It connects to thermal receptors in the dermis, and detects changes in surrounding blood to make decisions of whether to stimulate internal heat production or to stimulate evaporation. There are two main types of stresses that can be experienced due to extreme environmental temperatures: heat stress and cold stress. Heat stress is physiologically combated in four ways: radiation, conduction, convection, and evaporation. Cold stress is physiologically combated by shivering, accumulation of body fat, circulatory adaptations that provide an efficient transfer of heat to the epidermis, and increased blood flow to the extremities. There is one part of the body fully equipped to deal with cold stress. The respiratory system protects itself against damage by warming the incoming air to 80 to 90 degrees Fahrenheit before it reaches the bronchi. This means that not even the most frigid of temperatures can damage the respiratory tract. In both types of temperature-related stress, it is important to remain well hydrated. Hydration reduces cardiovascular strain, enhances the ability of energy processes to occur, and reduces feelings of exhaustion. Altitude. Extreme temperatures are not the only obstacles that humans face. High altitudes also pose serious physiological challenges on the body. Some of these effects are reduced arterial, the rebalancing of the acid-base content in body fluids, increased hemoglobin, increased RBC synthesis, enhanced circulation, and increased levels of the glycolysis byproduct 2,3-diphosphoglycerate, which promotes offloading of O2 by hemoglobin in the hypoxic tissues. 27. Environmental factors can play a huge role in the human body's fight for homeostasis. However, humans have found ways to adapt, both physiologically and tangibly. Scientists. George A. Bartholomew, 1919–2006, was a founder of animal physiological ecology. He served on the faculty at UCLA from 1947 to 1989, and almost 1,200 individuals can trace their academic lineages to him. 28. Knut Schmidt-Nielsen, 1915–2007, was also an important contributor to this specific scientific field as well as comparative physiology. 29. Ecology portal. Biology portal. Starting out in the field of zoology with a PhD from University of Rochester, 1933, Rand began teaching physiology at the University of Rochester in 1941. It is there that he partnered with Wallace Ofen to publish a graphical analysis of the respiratory gas exchange in 1955. This paper included the landmark O2 CO2 diagram, which formed the basis for much of Rand's future work. Rand's research into applications of this diagram led to the development of aerospace medicine and advancements in hyperbaric breathing and high-altitude respiration. Starting out in the field of zoology with a PhD from University of Rochester, 1933, Rand began teaching physiology at the University of Rochester in 1941. It is there that he partnered with Wallace Ofen to publish a graphical analysis of the respiratory gas exchange in 1955. This paper included the landmark O2 CO2 diagram, which formed the basis for much of Rand's future work. Rand's research into applications of this diagram led to the development of aerospace medicine and advancements in hyperbaric breathing and high-altitude respiration. Rand later joined the University at Buffalo in 1956 as the Lawrence D. Bell Professor and Chairman of the Department of Physiology. As chairman, Rand surrounded himself with outstanding faculty and made the university an international research center in environmental physiology. 29. Ecophysiology. Ecophysiology, from Greek oikos, oikos, household physis, physis, nature, origin, and logia, logia. Environmental physiology or physiological ecology is a biological discipline that studies the response of an organism's physiology to environmental conditions. It is closely related to comparative physiology and evolutionary physiology. Ernst Haeckel's coinage bionomy is sometimes employed as a synonym. 1. Plants. Plant ecophysiology is concerned largely with two topics: mechanisms, how plants sense and respond to environmental change, and scaling or integration, how the responses to highly variable conditions, for example, gradients from full sunlight to 95% shade within tree canopies, are coordinated with one another, and how their collective effect on plant growth and gas exchange can be understood on this basis. In many cases, animals are able to escape unfavorable and changing environmental factors such as heat, cold, drought or floods, while plants are unable to move away and therefore must endure the adverse conditions or perish. Animals go places, plants grow places. Plants are therefore phenotypically plastic and have an impressive array of genes that aid in acclimating to changing conditions. It is hypothesized that this large number of genes can be partly explained by plant species' need to live in a wider range of conditions. Light. Light is the food of plants, i.e. the form of energy that plants use to build themselves and reproduce. The organs harvesting light in plants are leaves, and the process through which light is converted into biomass is photosynthesis. The response of photosynthesis to light is called light response curve of net photosynthesis PI curve. The shape is typically described by a non-rectangular hyperbola. Three quantities of the light response curve are particularly useful in characterizing a plant's response to light intensities. The inclined asymptote has a positive slope representing the efficiency of light use, and is called quantum efficiency. The x-intercept is the light intensity at which biochemical assimilation, gross assimilation, balances leaf respiration,